Thanks for coming out today, you guys. I didn't realize in the university calendar I checked for conflicts with university holidays, but I just found out today that this afternoon is the engineering fall convocation. So thanks for joining us here today. And I heard from several people that they were unable to make it because of convocation. That explains the somewhat light turnout. If you could though, I do have the sign-up sheet up here, so before you go, please do sign in. And I think without any further ado, I'll introduce today's speakers. So Dr. Katinka Stecina completed her BSc in Biology and Chemistry at the University of North Carolina in Charlotte. And then she completed her PhD here at the University of Manitoba in the Spinal Cord Research Center, where she investigated the function of spinal neural networks in motor control. And then in 2006, she began her postdoctoral training at Gothenburg University in Sweden. And then in 2007, she was awarded a very prestigious Marie Curie Fellowship at the Copenhagen Neuronal Control of Movement Research Group. And I understand that's sort of a unique research environment where animal researchers and human researchers work very closely in parallel to understand the neuronal control of movement. So there, while she was in the laboratories of Dr. Holborn and Nielsen, she gained experience in many research methods while being part of that group. And through support from the Danish Agency of Independent Research from 2010 to 2014, working with Larry Jordan here again at the University of Manitoba, she's established an in vivo mouse preparation that's become the basis for much of her current research, including for some of her NSERC-funded projects that have just recently begun. So currently she's an assistant professor in the Department of Physiology and Pathophysiology, and I'm glad that we're able to get speakers from the Bannatyne campus down here every now and again and keep track of what we're doing all over the university and throughout the BME program. And so today she's going to tell us about some of the ongoing work in her lab.
Very excited to be here, meeting this year's thank you for wanting me to talk about my work. And just to put you in the context of what today's talk is going to be about, I wanted to show this simple slide of where my work pertains to. As much as I would like to, I understand not all of the biomedical engineering students deal with neurophysiology per se, and this is where I'm coming from. Most of the work you're going to hear from me today pertains to looking at the neuronal control of movement. And I have been doing a little bit of research in connection to these four areas. I started with looking at kind of simple reflexive movements and combined it with looking at how to understand reflexive versus more voluntary motor control. Then I like to think of my research as something applicable near the rehabilitation field of movement. So you're going to be hearing me talking about things in these four contexts. And most of all, however, I'm still closest to just playing around with spinal neural networks and trying to figure out how they work in a healthy, unperturbed context. So we know the nervous system matures, neurons are there, they develop a position and a role. Of course, with synaptic pruning, the function of the neurons changes a little bit, but it's kind of a simple system in a way. There's not a whole lot of changes throughout your life in it, and understanding how healthy circuits function is a way to help those researchers work on regenerating injured neural circuits. And this work also relates to understanding how, if healthy changes, is there a plasticity in the network? What kind of plasticity did we observe? And how the spinal cord may parallel some of the well-known brain plasticity networks. And the two objectives that I set out for today is really to give you some underlying motivation and examples of how to study some spinal sets of neurons begin by appreciating several methods that we use, mainly electrophysiological methods, to understand neuronal function. And please just feel free to stop with questions in the middle of the talk or anywhere during the talk. Okay. So just to bring everybody to the same level of where we are today, here is a very basic neuroanatomy picture from you. I don't think it's surprising to any one of you that in the context of motor and sensory motor function, I will be talking about something like the motor cortex that's organized in a very nice way that allows us special, easy kind of way to stimulate certain structures if we want to elicit voluntary movement in arms, in legs, or different places in the body. This motor cortex is well known to interact with the spinal neurons, which are really the final output generators of motor activity that live in the spinal cord, by the pyramidal tract system, which represents neurons originating in the motor cortex, having their axons descending all the way through the reticular formation via a depassation to the spinal level. Now, this gives us a way to examine how the voluntary control works. In the context of spinal or reflexive or more simplified movements, if you will, we have to look at a different type of anatomy shown here. The spinal cord is organized in a way that the dorsal surface of the cord contains incoming or afferent fibers bringing in sensory information. Whatever comes in the spinal cord is going through neuronal relays or perhaps going directly to the motor neurons, which give rise to the axons projecting through the mixed fiber nerves first, all the way down to muscle fibers. And electrical stimulation is one way that gives us an artificial way of activating these structures. Obviously, at different points, you're activating different types. For example, if you could just stimulate the sensory roots by applying your stimulation naturally at this point, you could separate effects from sensory versus the situation when you have a nerve that you're stimulating, which we typically use in human studies, non-invasive human studies. So this is a diagram just here showing you an example of what happens when you apply electrical stimulation at a nerve somewhere between the muscle and the spinal cord. It excites fibers in a way that fibers bring excitatory input back to the muscle, and this process happens in what's called an antidromic fashion, and you elicit an end wave first. And then there is also a second response that is the result of the excitatory input making its way to the spinal cord, being relayed to the motor neurons on the other, and then coming back again back to the muscle. Because with our human surface EMG recordings, we are taking a sample of activity of some muscle fibers just under the electrodes. Any questions so far? And this way of electrical stimulation is really a general way, and again, where you apply the stimulation at different points is what you can play with and give you the real options of looking at the nervous system. Now, here I have an example of a not too recent but not too far away either, from 2011. This is an article on one relatively famous Canadian neurorehabilitation specialist who works with functional electrical stimulation of arm muscles. So that means you're applying the electrical stimulus to the muscle fibers directly. And at this time, when the studies came out from his lab around that time, these studies have shown that taking people who have been injured, spinal cord injured patients, and lost the function of upper limbs and lower limbs, applying just muscle stimulation for a continued period was able to bring back some voluntary function, which is really, for the community of neuronal rehabilitation, this is a rather novel bite to swallow, because we know that in those patients who have suffered serious neurological injury, either at the level of the spinal cord or the stroke patient, it takes extreme efforts of traditional neurorehabilitation, and we still are not able to get them a voluntary motor output in most of the cases. And around about at the same time, or just maybe a little bit half a year or so later, there was a study in Lancet showing that in the context again of spinal cord injury and taking injured patients who were unable to move at all, so they were considered complete motor lesion, but what's in the actual tissue is always a different matter of what you are able or not able to do. But putting wires and tiny electrodes inside the spinal cord epidurally in these patients. At this time they were experimenting with 4 leads only. They were able to apply the stimulation, and after some point the subject was able to move a toe. So that's one of your most distal muscles, and with stimulation again, voluntary activity in a toe muscle came back. These studies were different in a sense, but also similar. If you look at where the stimulation was, of course they were different, it's hand function versus leg function, but they're similar because the underlying message is that, hey, if we do something drastic with these spinal neurons in 1 minute, if we buzz them so much for some period of time that they change, we can actually take the nervous system and allow it to interpret descending brain-induced signals with what's going on in your body, and maybe those two, what allows you to eventually move your toe after a long period of inability. So now, this is in the spinal cord, there are lots of neurons. And of course, putting functional electrical stimulation is not so bad. I don't know how many of you experienced it, but it's rather nice. Having 4 wires or 4 little electrodes inside your spinal cord is a bit more invasive, we should say. And of course, we have that group who was initiating those studies now, and all over the patients in this series, and they really sat on it. And of course, patients are interested. They're trying to do anything they can to improve. However, we still need to do a better job in understanding what is it that we really need to, or have to, or should do in order to improve this much functionality. Is it only really the electrical stimulation right inside the tissue that can give us so much advances? And understanding how and what each neuron does in the spinal cord is really the key for improving any type of intervention, such as these two. And the spinal neurons and how to describe their function is conceptually simple. You need to find those neurons that are activated by the brain. They also listen to some sensory input and connect different motor-related neurons in the cord and have some say-so in the final motor output. And the spinal cord is very different from the brain when neurons are organized often in 9 structures. You can outline the hippocampus and the layers of neurons or cortical neurons. It's function, anatomy. There is quite a good match. Spinal cord is a different type of environment in that context, and that's why this conceptually simple approach is actually practically quite challenging. What we can say in general about the spinal cord, that in addition to the motor neurons, which are really the final output motor activity generators, there are other neurons, what that we often just call interneurons, and some of those interneurons are considered last order neurons. So some of them are directly control what the motor neuron population does. So if we understood the function of at least some of the last order clusters of neurons in the spinal cord, that's kind of working our way backwards, or bottom-up approach, if you will. It may help us interpreting what we need to give and where in terms of the spinal cord to improve motor output. So if you were to find a key neuron for motor output generation, some of the characteristics would be something like these couple points on this list. So you would really want to find some cells that can coordinate sensory input, not just your commands from the brain, because motor activity is not ever in isolation under natural circumstances. You need an integrator type of cell. It needs to integrate sensory input with descending motor input. It needs to contact the motor neurons directly. For example, those motor neurons that help me stand up are neural extensors and knee extensors, and they need to be active at the time when I'm wanting to stand up. And they also, these special neurons, they also need to be able to talk to my extensors, not just when I'm sleeping, but also when I'm standing. And their selective activation should be necessary for us to have some activity. One special class of these last order interneurons, unfortunately not one of those that actually excite my muscles, but rather inhibits it, is what's called a reciprocal inhibitory interneuron that I want to talk to you about. So this concept of reciprocal inhibition, it's very simple. Maybe my picture doesn't quite project that, but if you never heard of the concept, all you need to think of simply is if you're trying to do a movement, flex your arm, the opposite muscle or the antagonist muscle need to relax for you to do this. This is the reciprocity between the two is working in a way that allows you to do what you need to execute. And these interneurons, this picture pertains to ankle dorsiflexion is when you lift your toe upwards, and plantarflexion if you press your toes downwards. And it just depicts that same message. And when I'm thinking that I want to perform dorsiflexion, naturally the brain needs to enhance my muscles, the tibialis anterior, allowing the dorsiflexion, and it needs to also tell the interneurons, these one here reciprocal inhibitories, to suppress gastric activity during that time. It's the opposite situation when you're trying to do the plantar flexion. Now, how can we use this simple kind of communication between spinal neurons in the context of looking at network function and spinal cord function? Well, again, we take electrical stimulation of the nerves as one of our probe. And what we are trying to do here in this simple diagram, I'm showing you that here is the muscle. Here is the the axons of the motor neurons that innervate the muscle. And if you take the opposite, if you take the stimulation of the opposite muscle, then you will activate the inhibitory cell, and it's going to silence or reduce the activity of these motor neurons. So when we take a test stimulation, which is simply knowing their anatomy and how can I activate, for example, the soleus muscle, take your electrode and put it in the popliteal fossa in human and apply electrical stimulation in the tibial nerve branch, and you get age reflex component is shown here as an example of what happens. Now, if you apply this test and you condition this test reflex by something, I mentioned here that I want to condition it in a way that the soleus is inhibited. I need to stimulate the antagonist muscle, so it's another nerve. But really the choice of conditioning in this example happens to be the antagonist muscle. But conditioning a test by something, that can be a lot of things, and we'll come back to this point later. But here the conditioning I put the test as a blue response and the red as the condition response. It will be somewhat different from the blue. And in this case, you see that it got smaller in amplitude. So when you try to quantify what happens with the size, the amplitude of this age reflex, based on the conditioning you apply and express it as percent control of this blue, the control age reflex, this is the graph you get for Soleus conditioned by TA. First, on the x-axis, you see the millisecond time difference between the test and the conditioning stimulus. They're very small time intervals, 1 to 10 milliseconds. You can see quite a different situation at each interval. When you stimulate at only 2 milliseconds between the stimuli, there is a profound suppression of the output from the Soleus motor neurons. This is what we call reciprocal inhibition. And this is what allows you to flex and not fighting for flexing your arm as you are trying to flex it, or your leg. This is a phenomenon known for a long time. This is not the first one, not the first study showing this. I just grabbed their figure. And why is this good for us? Well, this just shows us some type of interaction between spinal neurons. In this case, the inhibitory interneurons and other muscle afferents and our muscle functions, right? So when we look at this in normal subject, we have seen that there is a suppression. But when people use this method to look at hemiplegic patients following spinal cord injury or paraplegic, or even multiple sclerosis patients, so very different disease groups, if you will. There are changes. There are profound changes in what you expect here. So you see, first of all, none of the colored groups show the same pattern as the black dots here. Okay? This is really the take-home message. In fact, you see in the hemiplegic patients, there is a big facilitation of this reciprocal inhibition. So what are we seeing here? We are seeing here that there is a change in the nervous system, and the normal communication between certain groups of neurons is not as it should be. Now, when we apply rehabilitation strategies to some of these patients, we can shift these curves, the red, the yellow, and the green, and shift it back towards the black pattern. And this is what tells us, is my therapy working? Is this a responder to gait training? So some of this, unfortunately, reciprocal inhibition is not yet one of those tests that people do routinely in a hospital with like nerve conduction tests or measuring something. It is only done in some of the frontier rehabilitation hospitals, but it is a very good indicator for following the effects of your therapy in some cases. And when again, this was just one of the interactions I picked to talk to you about in the context of the muscles and spinal neuronal integration. There is many others. I think most of you, when you think of spinal reflexes, yeah, there is a stretch reflex, yeah, there is some inhibitory interactions, and then what? Right? There is a lot more. There is this. Most of them are inhibitory. Some happens postsynaptically, some happens presynaptically. So again, varying timing between different tests and conditioning. Some happens between agonist and antagonist muscles. Some happens between two synergist muscles or homonymous muscles. So they are all ways for you to see into the functioning of a healthy spinal cord. The 1A, or the reciprocal inhibition, is, you probably, all of you ask, okay, sure, there are these 1 millisecond changes, but does it mean anything functional? Well, maybe Nielsen and his group in Copenhagen, they were just lucky with 1A inhibitory interneurons. I don't know, I don't think so, but when you think of the role of this reciprocal inhibition, it is good and bad, right? You can flex because you don't have to fight the opposing muscle. But what if you're a ballet dancer? What if you want to be standing like this all day? Do you need to activate just the flexor and just the extensor? No. Most of the movements require co-contractions in ballet movements, in other sports too. But the group in Copenhagen I'm coming from, they specifically studied ballet dancers versus other trained athletes. And in fact, they found that the reciprocal inhibition was the smallest of all the different athletes in ballet dancers. And of course, we also know that these interneurons in the spinal cord that I'm talking about, at least in animals, and some evidence also exists in humans, they also receive input from the brain. So of course, when you train and when you want to perform certain things and a lot of co-contractions, you can modify the synapses, you can change it. But it shows even with a simple spinal test what you have done to your nervous system over time. So now I'm gonna just switch topics a little bit and jump into another one of these spinal neuron to neuron interactions. And that's called, I'm calling it the cross inhibition. And that really comes from the reflex that you probably all remember again from basic biology, that when you get a nociceptive input on one side of your body, your pain receptors say step away from it, move away from it. On the other side of your body, you evoke what's called, if it's your legs, we're talking about especially, you evoke an extension or an extensor activity because you want to make sure you don't fall. Now, that just shows that there is left-right communication. And of course, every movement, almost every movement is there. You want that to be there in the nervous system. And one of the, in this case, the painful information excites your extensors on the other side. But are there any other cross interactions? And yes, there is quite many depending on what you stimulate and where. But what I want to talk to you about today is just an example of coordination between the hip and the ankle on the other side. So left-right coordination, we know awful little about it in humans. As we understand it today, comes from cat studies when people looked at the spinal cord and the brain spinal neural activity of cats that were walking on treadmill or pegs or under different conditions. In humans, we really have no appreciation of what is the circuit. Do you need your brain to do left-right activity? You probably all heard of these neural movements that we are all born with. Babies, they do everything with two hands at the same time. They want to reach for a key, they lift two hands and they reach for a key at first. Those neural movements are there. Sometimes they come back in patients after stroke, or often they come back. So mirror movements are thought of being more or less equal cortical drive to initiate that voluntary movement you are trying. Now, we know that babies, right after they are born, or way before the corticospinal tracts maturate, can perform air stepping. Air stepping is just like you're walking, requires flexion extension alternating on each side. Or, this is a baby, cannot walk yet. Look at the baby holding one hip completely under his trunk and the other leg extended, completely straight, and then performing some arm movements at the same time. There is left-right coordination. We don't necessarily need the cortex to do a lot of it, but what is really left-right coordination when you and I are standing and walking? This was really the motivating project for me when I started my postdoc in Copenhagen, and I try to, because there's a lot of literature from the cat neuroscience studies that your hip muscles controlling your hip have a lot of control of activity in other muscles, ipsilateral arteries. I try to take hip afferent or hip muscle or anything that is related to the hip and look for effects on the contralateral side of the body in healthy subjects. And this was a setup which required not just stimulation and looking at the effects right away, but also we decided to combine transcranial magnetic stimulation with these experiments, basically just to gain more detailed information about what we are actually seeing. So here you see a picture of how you can imagine the setup of these experiments. First of all, these studies were done in humans walking on a treadmill. That adds another level of complication, but it's not so important for what I'm trying to say about it today. But we apply transcranial magnetic stimulation or stimulation electrically of different nerves, including the femoral nerve on one side, and recorded the activity of a lot of other nerves in the leg and in the trunk on the opposite side, to try to understand what happens when we look for that right coordination. So what does transcranial magnetic stimulation brings here, and what it gave me as an advantage was the following. So probably most of you heard of how to use basically a magnetic field in motion inducing electrical stimulation of structures a certain distance away from this moving magnetic field. That's the use is for TMS in our heads. And essentially we activate with the moving electrical charges either neurons in the cortex or their axons. And that gives us, just like the age reflex we have seen, we stimulating a nerve with electrical stimulation, it gave us something, a test reflex to look at and follow and to understand what happens to it. And this is shown here. Now, instead of me using conditioning stimulation at an antagonist muscle on the same side of the body, I use conditioning stimulation at the femoral nerve on the opposite side of the body. And another test reflex was, of course, the soleus age reflex evoked by the left tibial nerve stimulation, just like I've shown you in the context of reciprocal inhibition. Again, it's nothing, the concept is not different from what I talked previously, that you expect a different, an altered test reflex by the conditioning, and you quantify the changes. It's a regression or facilitation. It depends on a lot of things, but that's what you're looking for, change in your test reflex. So here are actual data examples of when there was, on this side of the graph, on this side of the page, I'm showing age reflexes in soleus when they were applied just alone or together with the femoral induced conditioning. The gray shaded area shows the actual age reflex component of the reflex. And here I used a distance of 20 milliseconds between the TMS stimulation and my age reflex, my tibial nerve stimulation to age reflex. In the lower panel, you see this arrow bar is longer here because I used a 70 millisecond distance between the two. And what you can see here that my conditioning evoked not a lot of change in the first case, but in the second case there was a big facilitation of my age reflex due to this conditioning. On the opposite side of this page, I'm showing you what happened to the motor evoked potential. So when I elicited my test reflex with cortical stimulation, with the TMS. So here in the first example, again, not a lot of changes, but in the gray area, when there was longer distance between the two, there was a facilitatory effect. So when you do a lot of intervals and time window examinations of this is how they can be quantified, these effects. So I'm showing you in blue the age reflexes and in red the motor evoked potentials, how they changed as a result of conditioning. And on this axis, the positive numbers and the negative numbers show the net change of conditioning. So when the bars are going down almost to -4 or 5%, that means the conditioning a reduction in test reflex. And the other way, it's basically looking at the facilitation of my test reflex. And the summary data shows that at different intervals, something different is happening. So the 30 and the 40 millisecond versus the 60 shows a clear different effect. In one case, facilitation, and in the other, suppression. And what is happening here is that I'm looking at a person who is about to take a step, and I'm starting from when they are working on after heel strike. Then you load, and then you pick up the other leg. And we just go back one figure. If you look at the second trace in either panel here, you see that other than an artifact due to my stimulation, wherever, a little bit less when I do TMS because my stimulation is far away from my recording in the soleus muscle. There is no activity in the soleus. It doesn't need to be active. It's a step cycle. It starts to be silent a little bit and then become active. And this is the point here, about like 300 milliseconds after the heel strike, is when soleus starts to be active. And basically the intervals I'm using are very early on in the step cycle when soleus doesn't need to be active. My brain is telling my soleus, shut down. It's inhibiting anything I'm trying to do peripherally, it's suppressing it. But when I'm getting closer to the point when soleus almost needs to come on, because otherwise I'm going to fall over and not be able to finish my step, the brain stimulation facilitates this soleus motor neuron pool and tells them, okay, time to wake up. You're allowed to fire. You need to activate the extensors. So this is what I think I'm seeing in these graphs here. And one of the fundamental questions in this project was, okay, so do we use the brain for this or not? With the latency of these effects, you can get a good idea of what is happening, but unfortunately we saw these effects at a kind of gray zone, and we needed other ways of looking into, let us decide if you absolutely need the cortex, or can you do things just by sending the sensory input up somewhere into the reticular formation, and then things integrated there, and that input can come back and give us sensible information to the opposite side. So that was the next step when I used not only transcranial magnetic stimulation, but combined magnetic with electrical stimulation. So you're all engineers, and I hope it doesn't take much effort for you to understand what the differences are in the context of brain stimulation. So the magnetic stimulation, of course, there is moving charges as a result of the moving magnetic field, but it doesn't penetrate as deep as pure, good old electrical charge put onto your skull. Basically, that's what we use, because the layout of the brain structure in the homunculus, at the beginning perhaps you missed that, but the legs are a bit deeper than, for example, arm muscles. But regardless of that, also just the charge density and the excitability of myelinated axons versus somas is different in the brain. So whatever stimulation you put in, you are likely to excite the fibers first with electrical stimulation, and the magnetic stimulation activates the somas of the neurons that are in your brain in the motor cortex. So by using these two, somewhat, it's not a complete 100% clear-cut activation, but it's likely, and there is a timing difference of a couple milliseconds that you can use as your, like, is it really doing what I think I'm doing with my stimulus effect? And what it can tell you is when you activate cortical neurons and you are influencing the motor output by cortical neuronal changes in cortical neuronal excitability, you don't expect that to be the same when you are just activating the axons of those neurons. because if your stimulation is activating just the axons, that means that that input is taken one relay down and farther down along the CNS axis, and the integration of your stimulus and the sensory input happens not at the cortex. And in our case, we have, and when you only activate the axons, then if the cortex should be involved, then you shouldn't see that facilitation there. And that's what was my expectation when I started electrical stimulation in humans in this project. So unexpectedly, what we got as the results was that no matter where we stimulated, in the cortex or just the axons of these fibers, there was a facilitation, meaning that probably the integration happens below the level of the cortical neurons. So you don't need your brain to be able to tell your ankle extensor on one side to be active as you're walking around. So this is just a recap and a summary of what I've been trying to show you with the studies. This is how the experiments were done under subjects were walking on a treadmill. They've been rigged up with a special helmet, basically taking off the weight of the TMS coil. I think most of you maybe didn't know how heavy that structure is, just to be carrying it around is like you have another head. So let's try to make our study more realistic by taking off the weight of that coil we tried. And they were being, we used electrical stimulation on the different nerves, so they had a lot of wires moving with them and coming with them as they were walking on the treadmill. But basically, these studies allowed us to draw kind of a map of what happens when there is femoral nerve input, which is either flexors or knee extensors, generate that. Coming from the right side of the body, it's relayed somehow via these commissural neurons that project from one side of the cord to the other, and they interact with the motor neurons that influence soleus muscle on the opposite side of the body. And based on the results that I just presented to you, we think that the cortical neurons are not necessarily involved in this, even when we just activate their axons, which means anything below the reticular formation can be sufficient to mediate this coordination. So this pretty much ends the bulk of the talk on human electrophysiology tests to look at spinal network interactions. There is just a few points I want to make on where I really started from, and what I'm really doing with most of my time at the moment is looking at more directly, if you will, into neuronal connectivity in animal studies. So if you have a way into stimulating the brain somehow and sticking very small electrodes into the spinal cord, you can actually understand the activity of single individual spinal neurons and go back to the simple conceptual map I showed you at the very beginning. What is their function? What's the input? What's their output? How they change their firing? Are they changing their firing based on the task posing them out to? So these are the basic questions that my lab is trying to answer. And the preparation that I worked with in the past for a long time, and not just me, but since the time of Sherrington, the motor control person that probably most of you heard of from the turn of 1911, was some of his first publication on how decerebrated animals show movement and the generation of, for example, locomotion, which is a very regular pattern movement, can happen in those conditions. And this is the model that allows in the lab for us to look at motor function specifically and sensory motor integration. Because otherwise, if you anesthetize animals for research, they're sleeping there. It's extremely hard to wake up the nervous system enough to get a motor output without running into some ethical issues, right? However, when the the deeper brain nuclei goes through to the thalamus. So you end up with an organism that has no ability to integrate any type of sensory or pain signal as conscious animals can. There's nothing there to feel pain. This allows us ethically to use this kind of preparation and remove the anesthesia in this state and apply electrical stimulation or pharmacology to manipulate the nervous system and get the motor output we are trying to look at. This can also be done in rodents, and you also have to appreciate that it's more than just looking at the nervous system. These living preparations. You need to monitor their physiological state. Just like in the hospital, you're doing surgery on someone, you have to pay attention to a lot of other things, including temperature monitoring is very, very important for rodents. End-tidal carbon dioxide, how they're breathing. We control their breathing the majority of the time with ventilators. We look at heart rate. We often take blood pressure measurements one way or another. We use different stimulators, electrical stimulation. We use amplifiers for taking nerve recordings from the hinding nerves or whatever nerve we are trying to understand in the context of the motor function. And the brain stimulation can also be added on top of all this if you want to truly look at voluntary versus reflexive control. And this is the mouse preparation is the one that I really hope to use in the future. Fortunately, when you look at central nervous system actions, cell-to-cell communication, which means in case of a neuron, action potential generated, synaptic delay, and there is another action potential in another neuron. Inhibitory or excitatory action potential, it doesn't matter, but that's the same in a human, and that's the same in a cat. 1 millisecond of synaptic delay by rule, in the spinal cord at least, is true in the human, in the cat, in the mouse, whatever I'm looking at. Not when it comes to nerve stimulation. Timescales change, as you will appreciate when it comes to looking at peripheral actions. Distances are shorter. Nerves are the same, more or less, as far as we know, but distances are shorter and timing is different. But when I talk really about the cellular, single cell level circuit, they're actually quite comparable, and you are at the same baseline because the neural processes of the brain are the same in the central nervous system. So many of you, or most of you, remember that when you are inside a cell, within the spinal cord, for example, if there is an excitatory input that increases the memory input, depolarizes the memory potential, brings it to more positive, close to a level where the cell can actually fire an action potential, and the opposite happens when there is inhibition coming from other neurons. This is just a little reminder I wanted to bring up before showing you some experimental slides. In these experiments, what we have done is trying to basically identify lumbar neurons in the spinal cord that are basically the equivalent of the human research I was just trying to tell you. And so where are the neurons that control my ankle extensors when there is an input from the hip? Okay? And this is an example of what we can do. We use very small, around 5 micrometer or less diameter, custom-made glass micro-electrodes that are filled with a conductive solution, and take also a fluorescent dye and add it to this solution. So when we go and look at intracellularly the activity of a neuron, we actually inject some of this dye into them. So here is an example when we inhale a small spinal neuron in the lumbar cord. We stimulate pyramidal tract up near the neck, and we get this nice excitatory potential that's going on. Right? We take another site in the brain. This happens to be in the reticular formation active during walking. This MLF pathway would be active during walking. And we look at the input of our cell, and sure enough, there is an even bigger excitation evoked in this cell by very small 150 micron stimulation. And then we use nerve stimulation in the hind to test, does this neuron also listen to input from the hip? This happens to listen to input from your hamstring muscles, because when we stimulated that electrically, it got very nice excitatory postsynaptic potentials. So this would be one of the candidates that happens to get input from the hamstring muscle, and we found some that projected, you cannot see this really well here, but some of these neurons were projecting not necessarily only on the same side of the spinal cord, but across the cord and terminating here on motor neurons. And when we looked at how the dye could be traced, so the dye goes into the soma of the cell, then it makes its way down the axons and out into the dendrites within a couple of hours. So you can follow what you labeled by sectioning the spinal cord, take it out after the experiment, fix it and section it into very fine sections, frozen, and then under fluorescent microscope you can always see where your little red dots are if the dye was red. And that's exactly what our colleagues reconstructed here, and also described how far the red is extended from this one particular neuron. So we have an appreciation of, okay, one cell, how far can it affect its neighbors in the cord? And also by using other markers, for example, the gafrin, which is a marker for an inhibitory neurotransmitter, we can look at whether our cell of interest was actually contacting spinal motor neurons, and those are labeled with blue dye here, cholinergic marker, and the little light blue gafrin puncta, so the little ending of the axon we followed, are close, very close to this spinal motor neuron. So they overlap with our dye, the gafrin puncta, and this big blue motor neuron is shown here, and the little portion of it is shown here, where these two arrows are. So this is how we kind of map the anatomy, try to connect it to function, but there is one more step, because these studies have been done in resting cats, sleeping, anesthetized animals. Now, are these neurons silenced when I'm walking, or they actually recruited during walking? Is kind of how you would make your way from resting to motor actions. And I think this is the advantage of switching to rodent models in these experiments, because it allows a little more specific activation of neurons, and I can evoke a fictive activity. So these animals are, yes, moving, but we put them under neuromuscular blockage, so all the muscles are inhibited, because we want our micro-electrodes in the spinal cord to stay in there and not to move, so we can follow neurons for a long time, or through signals. And we try to use animals that have been engineered either by using optogenetic markers, so generorhabsin is a membrane receptor which you shine a light on your cell, a certain wavelength light, and it activates this receptor and it starts a cascade of events that results in an action potential in your cell. So this is what really answered the BME funding for, and I was in the beginning of starting this kind of project. But I'm very excited because they're allowing a very, very fine, more precise than ever manipulation of spinal neurons, and really going to answer the question, do I need all the neurons from my lobar segment to do something before my motor output changes? Or do I really need just one little area, or somewhere maybe somewhere else where I didn't think of the neurons are so important for influencing my extensor output during standing? So this is where I'm going with this research. So in this very brief summary, I hope you have now an appreciation of what spinal reflex pathway phase could mean under the textbook and why they may be useful. And really, motor output is not just motor function, right? Motor output also represents your healthy brain's control on this side of the brain, right? It's not only cognitive changes that happen with Alzheimer's. It's not only cognitive changes that happen after the death of the dopaminergic neurons. It's the motor function changes too. So can we use these very subtle communications in the spinal cord to infer what is happening to a person maybe in very early stages of Alzheimer's or other diseases to understand more about the nervous system in general. And really, I need to acknowledge everybody in my past, especially in the university at Göteborg as William Koster and Hammer were collaborators on such animal work I showed you, and in Copenhagen in Denmark, Jens Boniussen, whom I worked with in the context of the human studies, and Hans Hallborn was really the founder of how to transfer reciprocal inhibitory network research from cats to humans. So, and here at the UFM, I received always a lot of support from David Curry and Larry Jordan and all the technicians and support staff in the Spinal Cord Research Center. And I'm happy to be taking your questions.
Yeah, we do have a minute or two for questions if anybody has. Yeah.
Thank you for the perfect presentation. I'm just wondering, did you try different shape of waveform simulation, for example, the square wave, or because most of the simulation signal that you use is just single frequency, and if we use, for example, triangle shape, or so, what happened then?
That's a very good question. Yes, I specifically talked about single pulses here today, but very much it means a very different thing to activate things repetitively. It's really, again, another way of combinations and permutations you can play with to get to the structures you want to examine. For example, on one of the slides mentions post-activation regression, which is another kind of process happening that specifically requires a certain set of frequency and a certain set of pulse durations. But yes, to stimulate a nerve, 0.2 millisecond pulse is more than enough electric. And we just, in all the results I talked to you today about pulses in the animal studies at 0.2 milliseconds, in humans, again, single pulse, very short and very good.
Any other questions? I'll maybe ask one because I was really interested actually right before you popped up this slide about the optogenetics. I was writing a question if anybody had tried optogenetics. So in terms of spinal cord preparations, have people done anything like this before? Is this totally novel?
Optogenetics?
Yeah, like optogenetics in the spinal cord, has this been done?
It's been done in young animals.
Okay.
And smaller circuits, but in the in vivo animals, it's just a couple other labs beginning to be doing this, but
Okay.
it hasn't taken mainstream.
Okay, yeah, that's really neat. And then I was going to ask, like, how far or how local do you plan to study these circuits? Could you do a complete laminectomy and basically walk up the spinal cord and record from different sites? Like, for example, if you wanted to see if it went up to reticular formation, would the plan be to stimulate somewhere lower and record all the way up that high?
It's really, each animal is a little bit different.
Okay.
We just worked out a method in the rats to look at all the way to the higher thoracic segments and
Okay.
cervical, and it's